My dear students of 12th class, and today in our botany we are going to discuss codominance, and we will know about the multiple alleles also. Now try to be attentive so that you may be able to grasp what I am going to deliver. And previously we have discussed the incomplete dominance, and we have also discussed the general crosses where the F1 generation resembled either of the two parents that indicates the dominance or clarifies that one of the genes in an allelic pair is dominant over the other. But here we are going to discuss codominance, wherein in an allelic pair both the genes are equally dominant. And also we may learn about the multiple alleles. And here we go. Previously we have discussed about the crosses where the F1 resembled either of the parents that I have already told you is the dominance in case of tall and dwarf plants which were crossed. The F1 generation produced all the plants which were tall. That indicates the dominance of the tallness factor. Out of this the character height, tall is dominant over the dwarf which is recessive, and either it was in between, that is incomplete dominance, which we studied in the cross of antherinum, wherein the red color flower was supposed to be dominant over the white color, but the result in the F1 hybrids, they were all pinkish in color, and that determines the incomplete dominance. That is, the red color flower was not completely dominant over the white color. But in the case of codominance, the F1 generation resembles both parents. A good example is different types of the red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by gene I. This is how we represent the gene I. That is, the blood grouping in the humans is controlled by the gene denoted by the capital I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of the sugar is controlled by the gene. The kind of the sugar that is found in the plasma membrane of the red blood cells is controlled by the gene. The gene I has three allelic forms that is represented by IA, IB, and small I. And the alleles IA and IB produce a slightly different form of the sugar, while the allele I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over the other I, small I. In other words, when IA and I, small I are present, only I express, because I does not produce any sugar, and when IB and small I are present, IB express. But when I capital A and I capital B are present together, they both express because they are equally dominant their own types of sugars, and this is because of the and it is called as codominance. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are 6 different combinations of these 3 alleles that are possible, and therefore a total of 6 different genotypes of the human ABO blood types how many phenotypes are possible, that can be comprehended from this table. Here in the top you can see allele from parent 1 and allele from parent 2, and these are the genotype of the offspring, and they are the blood types of the offsprings. These are tabulated in this. In total there are 7 different types of the blood groups: A, AB, and then again A and AB and BB and O. In general we have these are the types of offsprings that are possible by the combinations of the different genes. And now this ABO blood grouping also provides a good example of multiple alleles. These multiple alleles, this can be seen that there are more than 2, that is 3 alleles governing the same character, that is the blood group. Since in an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. That is when we study the blood groups of different people in a population, and we can have the clarification of this multiple allelism. Hope you understand this. Thanks. Have a nice day.